at this. Okay. I just got a prompt of the recording. Okay. Let me share my. Okay. As I think as Tran mentioned, that we study a few things around human immunity, and today what I'm going to talk about it's entitled within my most comfort zone. I will discuss the affinity maturation in the germinal center, but I do want to start from a few years back on how we started to look at this issue, and then set the stage for some more recent work on the TB interaction inside the germinal center. So I think for this audience, it really needs no introduction. The broad setup of the germinal center, as shown in this little video clip. The red cells, those are germinal center B cells that are undergoing hypermutation, and they through a process we call it the affinity-based selection, they will generate the memory cells and plasma cells that we want from the human immune response, and that underlies many of of a vaccine, at least the antibody-based vaccine, which is more relevant than ever today. And of course, when this process goes wrong, that can generate autoreactive cells and also oncogenic events leading to lymphoma. So this selection of the useful cells to be outputted from the germinal center is critical. And many cellular players are important for this process, but perhaps it can be argued the most important one is the T cells. That in a nutshell, it helps the B cells in the process and the specific recognition that we are all familiar with. And just to emphasize a few important points is that the B cells using their B cell receptor, which is not depicted in this model slide, that internalize antigen in the process of presenting the peptide MC. Then the T cells that can recognize this antigen presentation in the context of co-stimulation, then provide signals that B cell need to survive and proliferate. And among all the signals the T cell deliver, one of the most important one is the CD40 ligand that engages the CD40 receptor that generates many pro-survival and the differentiation signal to the B cells. So, but of course, the T cells are to be at this position to or in this stage to help the B cells require prior activation. And we were just talking about the in vivo tissue environment. So the T cells to get here into the germinal center to help the B cells actually it needs a long journey. So initially T cells live in the so-called T cell zone where this blue cells are. That's naive T cells are all here, and the red zone is the follicular area, and the center is the germinal center. So to get here from the T cell zone, of course, the T cell need to be initially activated, then acquire effector function, and also they have to change their positioning program. So to be in the T cell zone, they have to be the most important guidance molecule they express is CCR7. But to get to the germinal center, the follicle, the B cell need to operate with CXCR5. So the T cell, we know that when they are in the germinal center, we call them follicular helper T cells. And so that's a special subset. And we think that's, as a field, we feel they are endowed with a specialized helper function to promote the germinal center response. So a very intuitive model proposed many years ago, and also were still a prevailing view, I believe, is that the T cell activated by the dendritic cells receive a suite of differentiation signal from the APC. Among many of them is ICOSL stimulation, then turn on transcription program that governed by BCL6, the master transcription regulator, then promote a TFH development program that is characterized by CXCR5 abrogation, so that the cells can, through this process, acquire the ability to be in the GC. So this is one way to construct this how we get a helper T cell inside the germinal center the B cell need. So now it's almost like 8 years ago. So when we realized this is not the whole picture, because if in a simple-minded way of construct this is like linear instruction differentiation process. It's the ICOS, the co-stimulation signal, that turn on fate determining factor inside the T cells that then drive effector program that is defined by a molecule that's important for the T cell function. But if that's the whole story, then we should be able to rescue, say, ICOS deficiency with the downstream effector molecule. But that's actually not the case. So when T cell cannot express ICOS, then you cannot rescue these cells by overexpressing either BCL6 or CXCR5. When we measure the overall outcome of the human immune response by looking at their germinal center formation. So what it turns out happening is that there's this bystander interaction that we define. It's that activated T cells, when they not only need CXCR5 to guide their migration toward the germinal center, but they also need ICOS signal engaged by ICOS ligand on the non-antigen presenting cells, or mostly naive B cell inside the follicle, and that triggered the or constitutively trigger the PI3K activity inside the T cell that can optimize their migration, and so the T cell can migrate across this border into the follicle, then eventually end up in the germinal center. So if the T cell that have no ICOS expression, not only they cannot express CXCR5, but also even if they can express CXCR5, they cannot engage this bystander ensemble of B cell in this way to be able to migrate. So my slides, please. Okay. But this is a rather unconventional, because we typically think about T cell B cell interaction in an antigen specific manner. So I make a contrast between these two type of interaction. It's a bystander interaction, as I just mentioned. It is a one activated T cell going to the follicle, and they are immersed in the ensemble of naive B cells as a tissue environment. So, and that's the ICOS ligand presented by the naive B cell in that context is rather an environment that is persistent and inescapable for any T cells that be in that environment. So this bystander signaling is constant. On the other hand, the antigen-specific interaction, as we know, forming an immunological synapse between a T cell and B cell, it is one-on-one. So T cell is monogamous, and so the percentage of antigen-specific cell, even at the peak of immune response, is still a minority of all your T or B cells. So the encounter between antigen-specific T and B cells is rather sporadic. And so if we specifically think about ICOS ligand interaction, this co-signaling is not persistent, it's not constant. So to make this point, if I show you this image that become more apparent, is that if I label all the B cells inside the follicle, which is the red cells here, and the green cells is the T cells that have gone into the follicle, you can immediately appreciate that for any given green cells inside this red ensemble, this bystander B cell will be really an inescapable field. So these early work that give us the concept that actually, even though all the lymphocytes are migrating and together they actually form a tissue niche, and the molecule that it expresses has a very important implication for the T cells to develop in a particular functional way. So, and so subsequent work that we realized not only the ICOS ligand, the classic P7 family the costimulatory molecule can work in this bystander way, but also PD1 and PDL1, which is actually inhibitory to T cell motility. So the helper T cells that express both sets of co-stimulation actually use them in a bystander way to interact with this ensemble of naive B cells and this tissue niche to optimize their migration and helper function. So, but of course, this germinal center itself is still an antigen-specific B cell ensemble. So for a helper T cells to reside and to functionally help the B cell in this tissue environment, they need to have the right TCR specificity, and that signaling is also governed by co-stimulation, not only the classical B7 family, but also SAP/SLAM. And from a migration point of view, the T cells to go from a T zone into the follicle, then into the GC, they actually need to know where the germinal center is, and there are suites of molecules that help the T cells to understand or to sense the border of the germinal center. And one of them is a set of a neuronal guidance molecule in a semaphorin and the pexin family. So the T cells use the semaphorin to engage the pexin B2 that expressed by all the germinal center B cell to understand the germinal, where the germinal center is. And once they get into the germinal center, there's also they experience a niche regulation exerted by another set of neuronal guidance molecule that is defined by germinal center B cell expressed FMB1 and the T cell expressed EPHB6. So this is actually a very curious phenomenon that T cells, once they are activated and get into this tissue environment, they express EPH receptor that exert a repulsive effect on the T cell. So the T cell cannot stay inside the germinal center for too long, and once they get in, they experience this effect to be pushed out. So this is actually my personal view is that this is actually very hard to reconcile with a pure linear differentiation model, because this is a negative regulation that is exerting the environment and in response to acute exposure to these niche factor that provided by the ensemble of the germinal center B cells. But what's even more interesting is that what's the T cell in here, they actually need to help the B cell. As I mentioned, CD40 ligand is important for B cell to survive, differentiate, but also B cell needs cytokine to differentiate into eventually the plasma cells. And IL-21 is of course regulated by IL-21 production from the T cell. Of course, is regulated by TCR signaling, but also by other factors, and that include, interestingly enough, is FMB1 from the B cells here and the EPHB4 receptor on the TFH. So this affin system is set up example is they have a sort of a timer that through this repulsive mechanism to kick the T cell out. But before the T cells get out, they are asked to produce IL-21 through this FMB1 EPHB4 axis. But once the T cells move out from the germinal center, and then they start to, they're not, as far as they're not escaping from the follicular area, they will be experiencing a soluble focusing cue, as mainly characterized by Jason's sister's group, that this S1P gradient emanating from the edge of the follicle or the sinus will work on or engage S1PR2 receptor on these T cells and to focus them back into the germinal center. So this, all these niche factor that is surrounding the germinal center actually regulate the T cell in such a way to give us this this sort of bungee jump dynamic phenomenon. Is that the T cells will get into the follicle, then they will recruit into the germinal center in an antigen-specific manner due to they have to recognize antigen and they recognize the border. But then once they go in, they will be pushed out. And when they push out, then they will be focused back in. So this dynamic go in and come out, go in and come out, we believe it's a process to strike the right balance for the right number of T cell adjusted to the size of this ensemble of germinal center in order to have the right ratio between the antigen-specific T and B cells to be the most productive ratio for generating high affinity B cells, and which I will touch on later in the talk. Not touch on, but actually discuss in more detail. But let me dwell on this ICOS-ligand issue for a little bit more. So with this setup, then we realize this is all sort of tissue niche pattern formation at the tissue level, but specifically for the ICOS receptor and its ligand. We don't seem to understand then how that play a role between antigen-specific T and B cell. So we ask this question whether ICOS ligand on the antigen-specific B cell per se will be necessary to program some aspect of follicular helper T cell to support the germinal center. So to test this question, we want to set up a system where we can specifically manipulate the ICOS ligand expression on the antigen-specific B cells. So as I mentioned, the ICOS ligand on the naive B cells or bystander B cells is really important for helper T cell. And to differentiate these two factors, then we set up this experimental system using HEL-specific MD4 mice. All the B cells are recognizing pan-egg lysozyme, and all the B cells are wild type. They can express ICOS ligand. Then we adopt transfer ICOS ligand sufficient or deficient splenocytes that contain B cell, of course, into this animal. Then immunizes the animal with NPKH. So in this setup, if we measure NP-specific germinal center response, then that have to come from the transfer cell because none of the MD4 B cells will respond. But the follicular environment will be formed by the MD4 B cells, which do not respond to antigen, but they can express ICOS ligand. So in this way, we can specifically ask the role of ICOS ligand on the specific cells. So if we look at their germinal center formation, then we find there's actually no difference between the germinal center size measured by FACS or on tissue section. So apparently the expression of ICOS ligand via antigen-specific B cell that are forming the germinal center is not required for the germinal center formation. That's obviously it's not what people typically think. Then we went on to ask specifically in this setup, now can we see the effect of ICOS ligand when we block it? So using the same setup, then we let the germinal center form, then we infuse a blocking antibody against the ICOS ligand and to ask whether there is any repercussion on the germinal center formation. So the premise is if this indeed, as it suggests in the previous experiment, that the role of ICOS ligand for the germinal center formation is all rest on the naive B cells, then this blockade should give someone is turning on their mic. Maybe you can turn it off. So so we ask whether this blockade would have effect. So the result is shown here. So this is already after the germinal center form in a comparable way. Then we infuse the antibody for a week. So this is control antibody treatment. So the antigen-specific B cell genotype, whether they can or they cannot express ICOS ligand, they have similar size with germinal center. If you block with ICOS ligand, then we see a very significant reduction in the germinal center remaining. So that's what you can expect. Actually, people typically interpret this in the literature as co-stimulation between T and B cells is really important for either TFH formation or germinal center formation. Except that then when we look at this blockade in the absence of ICOS ligand expression by antigen-specific B cell, we can still see the blockade effect. That means really this difference that made by the ICOS ligand is entirely coming from the bystander ensemble of the B cells. So that tells us, this niche environment that formed by this bystander B cell that do not participate in the germinal center response in an antigen-specific manner actually provide a very important niche environment to facilitate the T cells to be in that TFH mode in order to help the germinal center. So I think that's an important concept I try to convey. But then, of course, that when we realize this is the case, then become even more sort of bugging that one has to ask whether there's any role for ICOSL ligand between antigen-specific T and B cells. After all, it's a ICOS receptor. It's clearly something that can synergistically work together with the TCR. So to specifically ask this question, now with the previous experiment that we set up a system that we can perfectly use to ask, is that we can use the ICOS ligand sufficient or deficient MD4 B cells now together with the OT2 cells transferred into a wild type B6 mice, because I have shown you that antigen-specific B cell precursor, they don't have to express ICOS ligand themselves in order to form a germinal center. Now we can have a system that antigen-specific B cells that can express ICOS ligand or cannot express ICOS ligand in the same system. And so that study led us to realize there's indeed a role played by ICOS specifically in the context and the specific interaction between T and B cells. So here is just showing you this example that we realize what happens is this antigen ICOS ligand expressed by B cells is helping them to antigen-specific B cell, helping them to recruit to allow the T cell interact with them in a very efficient way. So what I'm showing you here is the cell in the white color is the OT2 helper T cells. The green is the wild type antigen-specific B cells. The red is the ICOS ligand knockout antigen-specific B cells. If we play the movie, you start to see that the helper T cell will have a larger surface area when they interact with the B cells as compared to interacting with B cells that cannot express ICOS ligand. So that, historically, a very important study from Michael Halen's group many years ago that have shown in vitro that T cell B cell antigen-specific interaction involve T cell spend a large surface area grabbing the B cells when they recognize the antigen, and that coincide with the calcium flux, and indeed that actually require the calcium flux to maintain that configuration of T cell wrapping around the B cells. And we can recapitulate this in vivo by using a calcium indicator, as shown in this example, that the heat map actually shows the calcium flux in the T cells that are in green color, and the germinal center B cell is in red. What you see is that, if a T cell passing by a B cell, there's not much of a calcium, but if a T cell come in to grab this B cell in the mode that we term the entangled mode, when they wrap around, and there's a very strong calcium flux. So, and it turned out in vivo that we can see for B cell that can, antigen-specific B cell that cannot express ICOS ligand versus those that can express the ICOS ligand, the calcium flux that each time the B cells bump into the T cell to trigger, they have 3 to 5-fold of difference in terms of efficiency. So every time the antigen-specific B cell interact with the T cells, the ICOS ligand help the B cells to trigger a better calcium response in the T cells. So now at the single cell level that we can see in the context of antigen-specific recognition, ICOS ligand can enhance the calcium signaling in the T cell and increase their contact area. And that suggests that this co-stimulation would provide a better stimulation to the T cell and means the better help delivery. And at the time when we were looking at how the ICOS ligand expression is regulated on the germinal center B cell, because that become an important factor in this context. We realize specifically in the germinal center B cell, and specifically with the CD40 signal, the ICOS ligand expression in the mouse system will become upregulated. And this is in contrast to the germinal center B cell that are engaged by BCR, they actually downregulate ICOS ligand. And this is in contrast to non-germinal center B cell, they don't change their ICOS ligand expression very much with the CD40 or BCR signal. So there are apparently a specific linkage between the ICOS ligand expression by the B cells and the experience of CD40 signaling in those cells. And that gave us a hint of there's potentially intercellular positive feedback inside the germinal center that work in this manner. So T cells, helper T cells that are activated inside the germinal center, they will have CD40 ligand on their surface, and also have CD40 ligand inside the cells stored in the vesicle. So germinal center B cells comes in, even if they don't present any antigen, they can utilize the CD40 ligand, the little CD40 ligand on the T cell surface, because B cell constituted express CD40 receptor. But if the B cell present antigen, and in particular in the context of ICOS ligand co-stimulation, and I emphasize this is a true co-stimulation because now the ICOS ligand work together with the peptide engaging the TCR, and this ICOS receptor in the helper T cells will promote a better calcium response. The T cell would grab the B cells with a larger surface area, and more importantly, this calcium signal is driving the externalization of CD40 ligand to the surface, so that these particular B cells can utilize more of the CD40 ligand, and then that CD40 receptor send a positive signal to the B cell. Among other things, the B cell will upregulate ICOS ligand. So of course, this B cells will disengage from the T cells after a few minutes, but over time they will accumulate the ICOS ligand on their surface, and then this ICOS ligand will help this particular B cell and its progeny to interact with T cell more efficiently. You can imagine if the B cell by chance acquire higher affinity BCR, acquire more antigen, present more peptide C, then it will kick off this positive feedback loop to allow this particular B cell to be more efficient and positive selected. And that's indeed what we can see using experiment genetic preservation, is that if we specifically knock out ICOS ligand on the antigen specific B cells, well, to disable this positive feedback, then you can see over a period of several days, when in the wild type cells there is a very dramatic increase of affinity, the knockout germinal center B cell cannot increase their affinity very efficiently, simply because this positive feedback is disabled. So with that, then we realized through this series study that inside the germinal center, when T cell and the B cells are interacting with this very dynamic manner, the help from the T cells on individual basis is delivered more or less in sort of quantum manner. They spent the time together for 3 or 5 minutes each time, then the T cell deliver some help, and this help is being facilitated by the ICOS ligand interaction in this antigen specific manner, not so much by programming the TFH development, but by controlling this delivery and establish a positive feedback loop to promote the positive selection. So with that, then we get a fuller understanding of how ICOS ligand, in particular receptor ligand, is regulating the germinal center responses. In a bystander manner, it actually facilitates the TFH development and the maintenance of their phenotype. And in the context and specific recognition, it mainly to regulate the help delivery to positive select B cell clones. So now, if you remember my initial introduction of how we see TFH development as external signal or co-stimulation into internal BCL6 upregulation, then specify a developmental program. But if the ICOS actually doesn't so much program the TFH development, what about the function of BCL6? And actually, if you think about it, although it's universally observed that the BCL6 is absolutely required for TFH development. But actually, from a cell biological point of view, we understand quite a little, not so much, about the essential TFH function that BCL6 directly control. So we from this development, TFH development, we also wonder, whether we can somehow rescue the BCL6 deficiency in order to understand what's the downstream effect of the BCL6 control. So again, we did this simple-minded experiment by overexpression of the CXCR5 on the BCL6, which obviously is one aspect the BCL6 is required for, because the BCL6 knockout T cell really don't express CXCR5 several days into the immune response. And what I show you here is it's a BCL6 knockout, and they don't go into the follicle. And shown here, this is the green cells. The red area is the follicle. If we overexpress CXCR5, then the T cells can end up in the follicle. This is in contrast to the ICOS. As I mentioned earlier, if you knock out ICOS, then you overexpress CXCR5, they still don't end up in the follicle very efficiently. But here, BCL6 knockout, you overexpress CXCR5, they end up in the follicle. So you wonder, okay, so now the T cell can get in the follicle. Do they help B cells to form the germinal center? And the answer is no. So BCL6 knockout, when they overexpress CXCR5, the germinal center formation helped by these B cell is not anyway higher than a GFP vector control. So, and then the hypothesis was, okay, the BCL6 must be regulating something inside the follicle, other than, the positioning of the T cells. And of course, I don't know why my slide. So, Chuan, are we still on?
Yes, we
Hello?
are. Yes, we are.
Okay. Maybe there's
the internet just broken down.
Okay, so where did I?
If you show up your slides, I can tell you where. Yes, here.
Right. So
Yeah.
Okay. I'm sorry. Maybe there's yeah. I don't know why this is. Okay. So so our hypothesis was could the BCL6 regulate, again, help the T cell B cell interaction inside the follicle. Then to probe this process, then we actually used a heterozygous BCL6 knockout. What's interesting is that because it provides this convenience, is we knock out only one allele of BCL6, then these cells initially activation, they express perfect level of CXCR5. They can get into the follicle. So 3 or 4 days into the immune response, provide us an opportunity to measure their potential effect, but missing with just one copy of BCL6, what's the effect is. And what's interesting is that even though they express a similar level with CXCR5 as the wild type cells at the beginning, and they do not support the germinal center formation as efficient as the wild type cells. And over time, this is by 4 to 5 days in the MD4OT2 transfer system, then the T cells start to lose their CXCR5 and or they do not express as high levels of CXCR5 and PD1 as the wild type. So that give us a system where we can read out the functional consequence of missing one copy of BCL6 where the cells are inside the follicle. And we looked at many aspects of these T cells, and one thing that stood out is actually the calcium signal in the T cell. Using the system I just briefly mentioned in a part of the ICOSL ligand, so we can measure the in vivo calcium flux of the T cells interacting with the B cells. What you see here is this is the wild type cells when they wrap around this B cell, they start to flux calcium, and more often than not, when the T cell misses simply one copy for BCL6, you can see that this is sort of a dark background. They don't flux calcium very well, and quantitatively, that's indeed the case. With the wild type cells, they have better calcium response than the counterpart that misses one copy of BCL6. And of course, so the BCL6 regulate how T cells respond to the B cells. B cells derived present the antigen, and similar to previous what I showed you with the ICOS, without an optimal calcium signal, their entangled contact is impaired. The CD40 ligand delivery is impaired, and there is a repercussion on TFH maintenance because they do not stimulate the T cell without one copy of BCL6. They cannot help the B cells as efficiently, and then we can use the experimental system to tease apart the intrinsic effect of BCL6 on the T cells, expression of the TFH associated marker versus those effects that B cell provides to the T cells to maintain their TFH phenotype. So I'm not going to spend more time on this aspect, but just to briefly summarize that the BCL6, we think that there are specific aspect of and the specific T cell B cell interaction in the follicle is being controlled by the BCL6 as revealed by this heterozygous setup. That is quite advantageous because total knockout, they never get up into the follicle and they actually they have problem with proliferating. But with missing one copy, they actually can express CXCR5 to get into the right position and to reveal this a very specific aspect of T cell biology is being regulated by BCL6. And along this process, now I'm using this one observation to switch gear to tell you that the last part of the story is that when we look at their dynamic interaction, we realize a very interesting phenomenon is that when we compare the wild type and heterozygous knockout T cells helping the B cells, and by following the B cells, I highlight with this migration path of this particular B cells. And what you see is that these B cells, as they migrate, and they more often bump into the wild type T cells than the knockout or the heterozygous T cells. Or should put it the other way, is the wild type T cells are more often to seek this B cells than the heterozygous T cell were able to. So what's this? Because there are roughly equal number of the T cells of this two type in the same field. So this suggests the heterozygous T cells has a reduced ability to to come into contact with the B cells, suggesting there may be a guided process. So why is this important? Now, if you think back of what I present to you of the T cell B cell interaction inside the GC in order to select the affinity cells. So every time the T cell need to come into contact with this B cell to test whether that B cell present more antigen or not, but they disengage in 3 or 5 minutes. It will be advantageous if a T cells can tell a good B cells or a B cell with an enhanced affinity from not so good B cells from a distance, particularly in this chaotic environment where everybody is running pretty fast against everybody else, and each one of them can spend time together for only 3 or 5 minutes. If a T cells can sense B cells from a distance, particularly when the B cells have a higher affinity, then that will make the system more efficient. So that particular B cell 6 movie prompted us to or gave us actually the confidence that there must be something working like a chemo attraction to promote this TB interaction. And this is a particular when we realized the B cells regulate calcium signal in a T cell, because many, if not most, chemokine receptor inhibition, and that guided migration is depends on calcium signal triggered by the chemokine receptor. So we now a former graduate student, Liu, now a postdoc that initially took on the job to screen for. Basically, we hypothesized there must be a chemokine receptor system that promote the TB interaction inside the germinal center. Then we screen for the chemokine that expressed by the GC B cell, and in particular, because our hypothesis is that the better B cells would have a better ability to trigger to attract the T cells, and so we hypothesized that the helper factor from the T cell would promote that chemokine production. So we use a CD40 agonistic stimulation in vivo to treat the mice for 4 hours and take out the germinal center B cells. And what's striking at the time was that the GC B cell upregulated the CCL22 by 1,000-fold in 4 hours. And we know that the CCL22 can attract the T cells through the very well-established chemokine receptor CCR4. So that seems to that become our primary suspect that maybe CCL22 and CCR4 are working together to promote the TB interaction inside the GC. And to test that hypothesis, we created a CCR4 knockout animal. Then this is just immunized mice. If you look at the CCR4 expression using the CCR4 knockout as negative control, you can see activated T cells. If they are not TFH, this is the level of CCR4 expression. If they are TFH, they express higher level of CCR4. The literature has well-established description of a Treg expressing very high level for CCR4, which we can recapitulate. And TFR cells, follicular regulatory T cell, also express CCR4, and this is just the quantitation of their expression. But functionally, what's interesting is that a Treg, not follicular Treg, but just Treg, they do respond to the chemokine ligand of CCR4, which include not only CCL22 but also CCL17. So in the following, so we basically use the two chemokines mixed together. Then you can see the T cell have a specific migration across the transwell. Whereas if you just simply look at total CD4 cells, then they do not have much of an increase in migration. And TFH cells also respond to CCR4 by migrating toward the 17 and 22 chemokine ligand. But curiously, TFR cells, they do not. I have to confess, we do not fully understand why this is the case, but this seems to be reminiscent of many aspects of regulatory cells. They apparently have a machinery, but they signal in a different way as compared to conventionally activated T cells. But the fact that the TFR cells do not respond to 1722 despite their expression of CCR4 provide a very interesting scenario where in the follicle, this chemokine receptor system is specifically regulated helper T cell interaction with the antigen-specific B cell. And to look at this, so we actually did many imaging studies in the follicular phase, which is in the germinal center phase. I'm just going to give you one example when we visualize the T cell, B cells in this particular stage and environment of the immune response. So what you are looking at is two types of B cells. One is a double knockout MD4 B cell that cannot express CCL22 or 17 in blue, and the wild type MD4 cells that can express these chemokines in green. And helper T cells inside the TC is the OT2 cells in red. So what we are going to look at is whether these two types of B cells can attract a T cell, come into contact with them comparably or differently. So I highlight these two cells with the arrowhead, so you can, and whenever the T cell bump into the B cells, I will label that with a broken white line. So what you see here is that this is the wild type cell, and that's a knockout cell. So of course, I give you a quite a striking example, is that the knockout cells barely have any cells to visit, whereas the green cells, which is the wild type, have multiple T cells come into contact. And it is important to point out that this type of experiment always involve transgenic fluorescent protein labeled cells that is not the green and the blue cells are not entirely equal. Besides the chemokine knockout the wild type, so it's important to do a control experiment where the wild type B cells are expressing either CFP transgenic fluorescent protein or GFP in the same field. And so in this setting, the two colored B cells both can express CCL22 and 17. So now you don't see the difference between these two types of B cells, as the red T cells bump to them with comparable frequency. So quantitatively, of course, we have to normalize the number of T and B cell in that tissue volume that we visualize, and we have to go through multiple imaging sessions and multiple mice. But what it turned out is, because every time we have a wild type internal control, then we also have wild type mix versus double knockout wild type mix. Then we can see if the cells cannot express 22 and 17, the B cell have a much reduced ability to attract T cell to come into contact. And that is, of course, a precondition for the T cell to spend any surface area or in antigen specific interaction with the B cells. And we verified also that we can recapitulate this phenomenon by comparing CCR4 wild type and knockout TFH cells. And it is important to point out that we measure the contact duration between these two conditions, and we don't see a difference. So meaning this chemokine is really calculating the frequency with which the T cells come into contact with the B cells. So now there seems to be a possibility. Now there can be a help attraction based intercellular positive feedback. What I mean is that T cell will inside DC will come into contact with the antigen-specific B cell, and that B cell can express CCL22. And because I showed you that agonistic stimulation through CD40, they can upregulate CCL22, then a B cell that experiences a very good help with the T cells in the form of this entangled contact will probably be able to produce more CCL22. And that they indeed express more CCL22 as compared to their peers in this tissue environment, then that B cell will be dynamic, sort of highlighted, and there will be more T cells to visit these B cells, and so the B cell will be receive more attention and more likely to establish productive interaction with the T cell in any specific manner, and so then you have a full circle, and that particular B cell will be better appreciated by the helper T cell group inside the GC and be positive and select them more. And so if this is indeed the case, we can make several important predictions that you do not normally make without such a positive feedback, and we can test those predictions. One is that germ center B cells of higher affinity should obviously express more CCL22. And to test this, we take advantage of a BUN high transgenic B cell that express a high affinity receptor against the NP hapten. So then we use NP hapten to sustain these cells when they form the germ center. And you can see this is the level of NP binding. And in the polyclonal B6 system, that we can see the NP binding is more spread and is lower compared to BUN high affinity cells. But we can separate this polyclonal cell population into two parts: the ones that are reaching the level of BUN high and those that cannot reach the BUN high. And we can verify by sequencing their BCR to look at a signature mutation, a hypermutation called W33L, which confer a tenfold increase of affinity to NP from their germline configuration. So we can see the red gate, and that's the high affinity that has this level of a W33L mutation. It's much higher than the cells in the blue gate. So this red gate, indeed, it's higher than the blue gate cell. And their level of CCL22 expression measured by quantitative PCR is indeed much higher in the high affinity part. And the ICOS ligand serve as a positive control based on the previous study I mentioned earlier in the talk. So the second prediction is that the converse must be true. So there's affinity and the CCL22 level correlation. The cell that express high level of CCL22 should be of a higher affinity due to selection. And to test this, we created a knock-in reporter by knocking IRES-td Tomato cassette right behind the CCL22 locus, and then immunize these mice. And then we can clearly see in the GC there are TD tomato high cells in this case, and we can sort the TD tomato high with TD tomato low as a indicator of the level of CCL22, and to ask their affinity content. And so now we can use W33L frequency as a surrogate for their affinity. And indeed, we can see in the CCL22 high part, there's 90% W33L mutation, and the CCL22 low, there's only 63%. And it's important to point out that the total mutation rate in the red gates is actually lower than in the blue gates, which basically means this must be a result of selection, not because of this red cells spending more time in the GC and mutating more often and to get a higher affinity. So CCL22 high cells are of those higher affinity cells due to selection. And if we, then another prediction is if we manipulate the help delivered to the B cells, then we should see reduced help leads to CCL22 downregulation, and increased help leads to CCL22 upregulation in a quite acute manner. So to test this, we again resorted to this knock-in reporter by treating the immunized mice with CD40 ligand blockade antibody. So as shown here, this is just a B6 control, so we can use that to sort to set up the TD tomato positive gate, and we can use control antibody treat the CD40 ligand blockade, and we sort the TD tomato positive cells, which is the cells that normally would make a lot of CCL22. By blocking CD40 ligand for only 4 hours, this is the time that TD tomato wouldn't be degraded yet. So we sort this cell out. We can see there's almost more than 100-fold downregulation of CCL22 from the treated cells as compared to the control group. That tells us the CCL22 expression indeed is acutely responsive to the ongoing T cell help. In the converse experiment, if we infuse a peptide epitope the T cell helper T cell recognize, so everybody in the GC present more of the peptides to receive more help, and to do the same experiment, we can see within the same short period of time, the B cells, germ cell B cell, upregulate the CCL22 by more than 100-fold. Again, they tell us the CCL22 expression is indeed an indicator of the ongoing T cell help. So with that tight correlation between the CCL22 production and the affinity on the B cell and their dependence on T cell help in a very acute manner, then one can predict in the complete absence of CCL22-17, the B cell cannot attract the T cells very well. Then the affinity-based selection should still work based on more random process, but it should be slowed because there's not such a positive feedback anymore. And to test this, we create a bone marrow chimera using double knockout bone marrow versus mu-MT bone marrow, because the mu-MT cannot generate the B cells. So in this system, all the B cells in this group would be unable to produce 17 and 22. And if we look at their affinity maturation process in the wild type GC from day 13 to day 21 over a week period, this is the level of affinity maturation from 37% to 87% in the GC for W33L mutation. In the double knockout GC, they do mature from 47 to 67, but it's much slower compared to the wild type situation. That tells us that this chemokine, which is working in a soluble manner that to attract the cells, actually is required for efficient germinal center selection. Because germinal center output is dependent on germinal center selection, so we can indeed see the end product of the germinal center reaction, high affinity plasma cell in the bone marrow over time is reduced if the B cells cannot express 17 and 22 themselves. The final prediction is now if we force the B cell that cannot express 17 and 22 to compete with the wild type B cells, then these cells will be numerically disadvantaged because they cannot receive sufficient T cell help. But whoever in this group end up in a system still contribute toward the, let's say, the final product, the bone marrow plasma cell, then those cells should be of higher affinity because they really need that higher affinity to compensate the lack of attracting the T cell help by every time we engage the T cell to receive more help. And to test that, we created a 1-to-1 mixed bone marrow chimera using the knockout and the congenital marker CD45.1. If we look at day 21 bone marrow plasma cell compartment, we can indeed see a double knockout B cell contribute to less to this compartment. But if you look at the affinity content of the knockout cells, now these cells have a much higher affinity compared to the wild type cells inside the same animal or compared to a wild type mix in another cell. That basically says without 17 and 22 chemokine expression, these B cells are fractionally reduced and affinity compensated out from the GC. And the final part, then we also spent some effort to look at whether similar system would operate in a human germinal center. And this is what we can find. So in the human tonsillar germinal center TFH cells, they do express CCR4, and the B cells do respond, the human B cell do respond to CD40 engagement by upregulating CCL22. And by single cell RNA-seq, we're going to see in the human GC, those DC B cells that express CCL22 have a enhanced signature of CD40 signal as compared to those individual GC B cells that do not express CCL22 from the tonsil. So these data suggest that human TFH cells and the GC B cells probably have a similar positive feedback operating between them. So this system, we think in a dynamic environment of the German center where everybody have to interact with everybody else, but nobody can engage anybody for a very long time, this chaotic environment should have to give rise to a at least a probabilistic determinism in terms of outcome of generating memory and plasma cell of a higher affinity. And this transition it really depends on positive feedback that mediated by this CD40 ligand and CCL22 system. So we think, so if you think back at what I told you early in the talk of ICOS ligand intercellular positive feedback in a context-dependent manner, so we think this affinity selection in the GC environment is really based on quickly selecting those better cells through two positive feedback loops. And we think that's probably important in terms of how we think about the positive selection of in context of vaccine. Like HIV, we know the useful epitope is really CD4 binding site, the broadly neutralizing antibody, but our immune system generates lots of antibody of higher affinity against the sugar epitope and everywhere else on the spike, but the CD4 binding site. Perhaps it's because the system evolved to quickly select the affinity enhanced mutant through these positive feedback. But those precursor to the useful epitope, initially their affinity is lower, so they're being disadvantaged and they take a long time to write. So if there is in the future, if there's a way that we can blunt the positive feedback in this particular content, we might end up getting this useful antibody against the useful epitope early on if we can have a more targeted way of manipulating. But that's, of course, hand-waving speculation. So that's the end of my talk that I think for the last most recent story was done by Follicular, as I mentioned in the series, the work that I presented early on was done by also He Ping and Dan Liu and Pei Wen and many other students, and I received funding from Ministry of Science, Technology and Natural Science Foundation of China, and Gates Foundation, HHMI. Okay, thank you very much.
Okay, so thanks a lot, Hai, for the fascinating talk. Anyone have a question can raise the hand. I see Yangxin has a question about the role of Treg cells in the germinal center. Maybe Yangxin can specify the questions.
Well, I guess the Treg also express the receptor highly, and I was wondering whether they go there to regulate the B cell or regulate the help T cell or TFH. Any assay to, you have condition knockout those in the Treg as well, I see from your slide.
Yeah, we haven't like breed the FOXP3 crate to specifically look at the CCR4. So what I can say is that CCR4 for sure regulate the helper cell recruitment to the T cell, because the system that we set up, like OT2 cell, we force them. There would be no Treg that in our visualization experiment, but we do not exclude the possibility that the TFR cells may use the CCR4 under certain condition, because so far the transfer assays, we take out those cells and that's measured in ex vivo. So they left the mouse for 4 hours. It's interesting to consider whether that process is being regulated by BCL6. Maybe BCL6 is shutting down the CCR4 response in the Treg just for that period we don't know, but we can measure. We should be able to specifically measure this for Treg. But more generally, Treg do regulate helper TB interaction, but that's in a way that we're still trying to fully characterize. It's not in a stage that I feel comfortable to present the data, but we are investigating those aspects.
Okay, so Xu, you have questions?
Yeah, hey, hi. This is really beautiful talk. I have a couple questions. I'll ask the two, and then if we have time, I'll follow up with a couple others. Just for the first part of your talk, it's really interesting about the bystander effects of the B cells. Could you comment on more why in the ICOS knockout in the bystander B cells, the T cells fail to migrate into the germinal center?
Oh, why they do not? Yeah, because
even when you overexpress 6 or 4, 5.
Yeah, so it's so apparently I think it's because the ICOS is really important. The bystander mode actually activate the PUN10 delta very well. So the T cell, their pseudopod extrude. So basically in vivo they migrated not a adhesion dependent manner, but they squeeze. They have to have the cytoskeletal reorganization cycle to migrate. Okay, and that depends very much on the PI3K activity. So when you take out the ICOS and without taking out PD1, and the PD1 working in the bystander mode is to suppress the PUN10 activity. So you create a situation where you have a CXCR5 to have the sense of direction, but you simply do not have sufficient PI3K activity to power you through to really penetrate. So we actually, in that situation, if we take away the PD1, then we see a recovery of a level of recruitment, but it's still not fully. So suggesting there may be additional factor that suppress. So this is a very counterintuitive. It's like you need some cells to be here, but you have a lot of negative regulation. But that's basically I take it. I think this speaks to the way that a simple cell intrinsic differentiation is not a model that can fully capture the biology in this setting. Is that the cells is in a dynamic regulation. Is that they all. Yeah, you always have a positive and you have a negative way of in response to that particular environment. So I don't know whether I answer your question. Is the signaling aspect of how is the PI3K activity and on the surface it's being regulated by ICOS and PD1, and perhaps there's another inhibitory pathways. And that cannot be overcome by simply overexpressing a lot of CXCR5.
That's very interesting. A quick follow-up for that is, do you think if you artificially activating ICOS on T cells can allow them to penetrate in stiffer environment? Say, for example, if you have fibrosis on the surface of cancerous tumors, and then activating ICOS on T cells, maybe to penetrating through the barrier.
Yeah, that's what I sort of proposed a little bit, but we haven't really careful look at it. I think part of I can imagine part of the PD1 blockade for solid. There are certain tumors that really have a very dense PD1 ligands expression in the macrophage surrounding the tumor, and I think that's quite inhibitory to T cell migration. But granted, so far we're looking at CD4. I cannot say for sure that CD8 would work the same way because their kinetics of PD1 expression is also different. but I can imagine there are, if you have a density now. So follicle is quite unique in that they packed with the naive B cell, and all of them express ICOS ligand, at least in the mouse, and all of them express PD-L1, very high levels. So that created that T cell environment. So in the tumor, whether you have such a dense pack, it's I guess that might vary case by case, but if they do, then that would I think that will have that motility modulating effect, either promoting or inhibiting.
Well, that's fascinating. My second question is really interesting. Many cells have expressed multiple GPCRs, and in this case, seems like for the T cells to migrate into the center require at least 2 GPCRs, CCR5 and also CCR4. And many GPCR tend to converge on the same signaling downstream for migration regulation. And from a cell biology perspective, how do a cell can tell the difference when you have 2 different ligands but converging on the same downstreams when they're in such a heterogeneous environment?
Yeah, that's a great question. I mean, the short answer is I don't know all the full detail, but their ligand distribution is different, and their desensitization kinetics and profile will be different. So the CXCR5 is more like a homeostatic chemokine that is the cells will follow the gradient, and the receptor may desensitize slower or not easy to desensitize, so the cells will migrate across the tissue domain, measured in tens or even 100 microns, because they migrate from the T zone to the follicle. But there's an alternative way to construct the model is they only cross the border using that chemokine. Then they totally desensitized and they trapped in this environment. That's also possible. Okay, so nobody has really in vivo to tease apart this. But for the CCR4, their desensitization must be quite fast. There are actually literature reporting that. So the T cell use it to tell the difference within 1 or 2 cell diameter. So that actually imaging is that they pay attention to this. Then another factor is that in a tissue environment, many cells express decoy receptor and enzyme that degrade the chemokine. So that helps to create the very sharp gradient or boundary. So a cell, there are certain environments that it has a high concentration. The cells will come into here and pay attention to that. But there's So I think that's the way I think of it.
Thank you.
Okay, so
thank
next is Haopeng. Haopeng, are you there?
Yeah, I'm here. Hi. Very nice talk. I have a question, kind of follow Xu's question. So there's many co-stimulation molecule on the surface of T follicular cells, and ICOS is the one chosen by the natural selection to guide the T follicular cells function. So my question is, why is ICOS? Why is it so special? You kind of guide the ICOS function as carcine signaling, but as we know, CD28 is also able to boost the carcine, and its ligand is also expressed on the B cell, which is B7. Why they can't use CD28 instead of ICOS?
Sure. I think that of course they use CD28, but I think it's but that's an interesting question. We haven't looked whether CD80 or 86 expression acutely responds to CD40 signal. So what's unique about ICOS in the context of antigen-specific TB interaction inside the GC is the fact that ICOS ligand apparently responds to CD40 signal. So you receive more CD40 signal, you put out more ICOS ligand, so that this arm of co-stimulation become more dynamic. Okay, so rather than just everything else is not important. It's this one is dynamically regulated. So similarly, the chemokine is also the CCL22 is dynamically linked to the recent past history of amount of help they receive, and that way you create a circuit or logic circuit between the T and B cells. So there are many B cells, and this is the logic of the system used to pick up the one that is really good and to focus their help toward these cells. I think that's how I see it. Rather than this molecule have a very unique signaling pathways. Yeah.
I see. I got it. Okay.
Baochun?
Hi. A lot of insight into the GC reaction. Really love it. I have a question. You well know that the BCR affinity maturation would require the multiple round of the cycling of the B cell in the GC dark zone and the light zone. I wonder, whether the factor you studied, whether they had any impact on the cycling of the B cell in the dark zone and the light zone.
Sorry, you mean like whether this process happened in the light zone or dark zone?
I mean, for example, whether the ICOSL-driven deficient or BCL6, that whether they also affect the cycling of the GC B cell through the light zone and dark zone process.
I think it would, because the cycling of the germinal center B cell would depend on the cells that succeed in the previous round, where imagine the round of selection, right? So they be positive selected, then they are allowed to re-participate in the GC reaction. So if nobody gets selected, then the GC will shrink, because you basically the dark zone is spent. So all the cells come to the light zone, nobody in the dark zone, then the GC would shrink. So it's almost everything that change the affinity selection would impact on this cyclic process. But I wouldn't go as far as to say that specifically regulate the cells going from the light zone to the dark zone. At least we don't have evidence to say that's a direct consequence, rather than a consequence of the selection being changed.
Yeah, that's so. I mean, so whether that's a system to allow you to track the JCB cell traveling between the light zone and dark zone, because for the cell to cycle, they need also not only positive feedback, but also they need to turn off some signal, right? Absolutely.
If they don't turn off, they just go through the roof. That's the mathematics. Yes. For example, the ICOSL-driven expression it's being negatively regulated by BCR signal. So if you only engage the BCR, then you actually you try to shut off the ICOSL-driven expression in the GCB cell, at least in the mouse system. So yeah, I think there are negative process. And going back to your first part, it's like whether there's technology advancement to allow us to, globally see the cell, track the cells for their entire cycle from light zone, dark zone, and the back and forth. It's theoretically possible, but still a very technically challenging prospect right now. There are newer imaging techniques that have larger field and higher resolution in terms of space and time, with lower phototoxicity, that one can potentially do that. And we are attempting to do those. We are still, trying to.
Thank you very much. Yeah, that's very, very good. Yeah.
Chao.
Hey. Hello.
Hey.
Professor Chi. It's really amazing talk. I enjoyed it very much. So you mentioned in the last part about the HIV immunization. Actually, I think it's a very practical question that we know that HIV antibody are generally this more brown neutralizing antibody have higher levels of somatic mutation, which means the B cell undergoes more rounds of proliferation and a higher level of AI-induced mutations in their Fab region. So basically, but in reality, the people undergoes immunization, vaccination of HIV, their B cell have lower level of somatic mutation, and they exit germinal center quicker than we thought. So what do you think could be the potential way, a possible way to retain those B cells inside the germinal center to let them stay longer, or attract them back to the germinal center to undergo higher levels of mutation? What do you think?
Okay.
Is there like small molecule or inhibition or activation of cytokines, or what do you think could be the potential way? Thank you.
Yeah, so I think if I understand correctly, you're asking like the problem of broadly neutralizing antibody precursors is that they need a lot of mutations inside GC, but they don't. It's very hard for them to spend that amount of time just stay inside. So ideally, you would have a GC that do not export cells, but just let them stay there. And but what's the I think there might be ways of doing that, like preventing the cells from coming out, but the selection criteria, it's not easily manipulatable, because start spending there a lot of time. Do they then for sure will become a BNAb type of cells, or you have to guide with the antigen as I guess the current sort of a thinking is that you immunize one antigen and another antigen is looking more and more closer to the final sort of a BNAb epitope. Is that more important? Practically speaking, I don't know how to see that, but I think that generally to have a larger germinal center and preventing their or shifting the balance of their being exported versus their staying, I think there are potential way of doing that, but that is mainly depends on how much we understand the cells exporting mechanism, which we are working on. What cells, when do they cells make a decision, say like I leave the germinal center rather than I stay here? That's actually there are interesting aspect to it. Hopefully I can report to you in your future.
Thank you.
Yeah.
Okay. Enfu, go ahead. You need to
Muted. Okay, it was a beautiful talk, very rigorous and thorough. And so there was some earlier work suggesting that ICOS ligand also interact with CD28. So what's your thought about that? Is there any possibility that the CD28 pathway is also triggered and contribute to this? Yeah.
Sure. I think that we cannot rule that out because we spend lots of time that it's not by design from the beginning. It happens this way that we can set up system to take away the ICOS ligand from the B cells that are antigen specific, then to T cell product. But we couldn't do the converse on the T cell side. We couldn't really just take away the ICOS on antigen specific helper T cell that are going to become a TFH to do this, right? So I can right now I couldn't rule out that part is playing a role. The only thing that I can say is it's still fortunate that we could see the ICOS ligand expression is being tied into this feedback. So if it's engaging CD28, as Hao Peng probably also alluded to, is that then the CD20 signal is have a similar. In this context, the co-stimulation is really, if calcium is the most important thing, of course, the CD28 could still work. But in our hand, like when we use those T cells with the anti-CD3 and the anti-ICOS, we find that ICOS works remarkably efficient with the calcium signal compared to other co-stimulation. But that could be, the in vitro setup that we use. Whether in vivo, that's we don't have a way to test that.
Okay, thank you.
Okay, so I have one question. So just from the molecular base, you actually mentioned BCL6 heterozygote have this impact on the calcium influx during the TB interaction. So is that CD40 ligand dependent, or what's your idea about this calcium change in terms of the heterozygote BCL6?
Yeah, so we think we don't have a rescue experiment. We don't have a rescue experiment, but without one copy of BCL6, the T cell expression of the STIM1 and the PLC gamma is changed.
So it's a collection of co-stimulating molecule downregulation.
It's actually calcium signaling pathways. So if we do RNA-seq, that we can see that pathway is being enriched as differentially expressed genes between these two type helper cells. But I cannot say that's a direct regulation of BCL6. It is a transcription repressor. It's harder to study than, let's say, it's a transcription factor. And I think Shane Crotty's ChIP-seq data, that if you look at their data sets, in human systems, STIM1 is actually one gene that BCL6 bind to.
I see.
Yeah.
Okay. Okay, so if no one else have questions.
Which one? Can I ask one follow-up question of your about BCL6? Just learn that because it's a transcription repressor, and the connection for CXCR5 is seemed to be upregulating that expression, or upregulation depends on BCL6. And seems like the two genes you mentioned also, my guess is that in the heterozygous, their expression level was also lower than wild type. Right. And what's your thoughts on how this transcription repressor eventually leads to the gene expression in these cases? Or is sort of a directly transcription network regulation, and potentially BCL6 regulating some other process eventually leads to down regulation of these class of genes?
Yeah, that's really I think that's a difficult question to. I really don't have a precise answer yet. My thinking is that the repressor depends on the protein that can bind to the locus together. It might turn into an activator, and at least rare occasions. That's one thing. Another is that they can repress transcription factor. Then that is the repressor again. So repressor of the repressor, which is what's being considered. But I find that it depends on the kinetics. We don't have the full kinetics yet. It's a if a gene is being regulated very fast, I find that a repressor seems to be harder to understand, because you have to allow the other repressor to be decayed into such a condition to turn it on. That take probably take a long time. I don't have more specific information, but these two are possible scenario. Because I don't have time, actually, our investigation of the BCL6 is not so much from a perspective molecularly the gene that it regulates, but rather to try to understand the cell, because BCL6 is so absolutely required, but very few other things are absolutely required. You knock out CXCR5, you barely have twofold the difference in terms of germinal center. You knock out BCL6 from T cell, you have absolute zero. And there are three factors that you knock out that you have absolute, or in a clean SPF mice, you have absolute zero. That is a BCL6, SAP, and ICOS. And I think it's not a coincidence that all the three, in our view, it's regulating T cell, B cell, antigen specific T cell, B cell interactions. It's because the effector molecule that you knock out, then you absolutely lose the germinal center, is CD40 ligand. So we think all of them are actually regulating how this CD40 ligand is being delivered to the B cell in a different sort of spatial temporal kinetic context of in vivo immune response. Yeah, so we are still at that stage. It's at the tissue physiological level to see whether we can reconcile this molecular players. They seem to converge on this actually quite simple cellular process. It's T cell seeing an antigen, then provide B cell help, then B cell go proliferate, then they be selected. So these factors are fundamental because they regulate this delivery process. But of course, the next step would be to understand whether it's a unique signaling pathway. But I suspect actually there's not so much unique. What we discussed so far is ICOS regulating calcium, SAP regulating proximal TCR signal, BCL6 now can, at least in our hand, regulate calcium signal. Then none of this is really specific to TFH, but they are put into this context of helping B cell then become a human immune. It's just so important for human immunity. I think that's how I see it, and that's what I want to propose.
Thank you.
Okay, so there are no other questions. Thank you very much, Hai, for the great talk. And then there's long discussion. It's the longest so far. Obviously, everybody is exciting. So thank you so much. So hopefully we'll see each other soon, face to face, in person.
Oh yeah, I hope so.
Yeah.
I hope so. You're all vaccinated. You are vaccinated.
Yeah, we all get vaccinated.
你们现在已经能回